Now let's take a look at the genome, the viral genome that is in the particle. Again, up on the upper left, there's our retrovirion, and I've been showing it to you with 2 green molecules in it. And these are 2 viral genomes, 2 viral RNAs. So every retrovirus particle has 2 of them in it. As you can see here, they're both coated with protein, nucleocapsid protein, and there's also reverse transcriptase in them, of course, and integrase and RNase H. This is what the genome looks like. Here's the 5' end here. It's capped. 3' end is polyadenylated. And it's interesting that this is coated with nucleocapsid protein because most of the plus-strand RNA viruses come in naked, remember? They don't have to be coated with anything because they can be translated as soon as they come in the cell. But this is an example of a plus-strand RNA genome that's not a message because this is not translated when it comes in the cell. This is going to be reverse transcribed. So it's coated with protein in order to facilitate that step. So the genomes are roughly base paired near the 5' end. There is some homology which allows base pairing between the same polarity. I know it sounds weird. This is not plus and minus strands. It's 2 plus strands, but there is a little bit of base pairing possible. It's not perfect, obviously. And then, of course, the coding regions are shown here for the various genes, the GAG, the structural proteins, the polymerase, and the envelope glycoproteins. All right, so in the virion, you could say the virions are diploid if you want, because they have 2 copies of the genome. Now, also present in the virion are 2 tRNAs, 2 transfer RNA molecules. And you'll see what the function of those are in a moment. These, of course, are derived from the host cell. They're not encoded by the virus at all. They are taken up during assembly by the virus particle from the cell, from the cellular pools. Now, we think that the genomes are dimers in order to give this virus genetic flexibility. These viruses are pretty resistant to UV and ionizing radiation. Most viruses, depending on the genome size, can be very sensitive to these treatments which damage the genome because there's no easy way to fix it. But we think retroviruses have 2 copies to make them resistant to these kinds of insults, which they will encounter, of course, during their movement within and outside of organisms. So they have 2 copies of every gene. So what we think happens is during reverse transcription, which we'll talk about in a minute, the enzyme is always switching back and forth between one template and the other. It just does so randomly. And so in the end, you increase the chance that you're going to get rid of deleterious mutations. So let's say the genomes have a mixture of mutations caused by UV or ionizing radiation. When the polymerase just switches back and forth, eventually by chance it's going to make a copy that's intact. And of course that's the only copy that will multiply. It may have some changes in it, but it will be viable. Okay, so that's why we think that these genomes, these viruses have 2 copies of all genes to make them resistant to these insults. So the mechanism that creates an infectious genome is called copy choice. It's the same mechanism that causes recombination among RNA viruses. Polymerase is copying one strand, and because the 2 strands are close to each other, it can readily switch to the other. And again, it does this probably a lot. Very often, it probably goes a few bases and switches back and forth, and it does it randomly. Okay, so let's take a look at how reverse transcription works. So at the top again is our dimeric genome, 2 RNA molecules slightly base paired at the 5' end. And let's take a look at just the very 5' end where we have this tRNA. So this tRNA is actually base paired to the viral RNA. So tRNA here, it binds to a place called the PBS, the primer binding site. So this is going to serve as a primer for reverse transcription. So it's an RNA primer that's going to prime DNA synthesis by RT. This part of the genome is quite interesting. You'll see there are lots of letters for different motifs. There are inverted repeated sequences here, IRS, IRL. And there's also a very interesting constellation here called R, U5, and 3'. And those will be, the importance of those will come up in a moment. So here on the bottom is the tRNA. This happens to be a tryptophan encoding tRNA, but different retroviruses encapsulate different kinds of tRNAs. And the way this is actually present in the particle is this is base paired to a rather complex structure. So without the tRNA, if you just took the viral RNA, it would fold like this in this region right here. So you can see the primer binding site is right here. We have the U5, IRS, IRL down here. But when you add tRNA, the tRNA melts apart this structure and fits itself in to bind to the primer binding site. All right, so this is basically a stem loop structure, and the middle part has the tRNA base paired very specifically, and there's a free 3' hydroxyl right here, and that's going to be the priming site for reverse transcription. All right, so that's the primer, and it's very specifically bound here. This is really neat that the virus is now utilizing a primer for reverse transcription, which is a host tRNA. I think that's amazing, and it evolved that way, obviously. So now we're going to go through this scheme of reverse transcription, which I think is really amazing. It's a bit baroque, but it has a means to the end. Its means is to make a proviral DNA, as you will see. So we start out with the viral genome in the capsid. It's dimeric, but we're going to just show one for simplicity. But remember, the enzyme is probably switching back and forth between the two RNAs. It comes in with the virion, with the tRNA, of course, base paired. And look where it is. It's very close to the 5' end of the genome, right? So the priming event, whenever it occurs, doesn't go very far, or so it would seem. So the reverse transcriptase uses the tRNA as a primer and begins to make the negative strand of DNA. So it's a light blue color to show that it's a negative strand. All right? So that is bound covalently to the tRNA primer, and the reverse transcriptase goes, of course, it reaches the end, the 5' end of the genome, and can't really go anymore because there's nothing left to copy. As the RT is producing that DNA, it's degrading the other strand, which is RNA, of course. So that's shown by the little pieces there of that green RNA. That little piece of DNA is called negative strong stop DNA, and that's only because when people were studying this, they would run gels of the product, and they would see this small piece of DNA, and they called it minus strand strong stop, and the name has stuck. It turns out now, in order for this reverse transcription to continue, this sequence base pairs with the very 3' end of the viral RNA. So here's the 3' end. It's a polyadenylated 3' end, and the sequence here is now complementary to that negative strand of DNA. If you look at the original genome, we have an R sequence at both ends, the 5' end and at the 3' end. And we have a U3 at the 3' end and a U5 at the 5' end. Those are different. And then, of course, the primer binding site. And so when you make this strong stop minus DNA, that's going to be complementary to the R because that's a copy of the R here. So this actually is going to now prime at the 3' end and continue polymerization. And remember, all this is in the cytoplasm, in the subviral particle. So here now we have that little piece of minus strong stop DNA. It's annealing to the 3' R sequence on the viral genome. That's called a template exchange. That's the first one because there's going to be another one, because now the DNA goes from this part of the template to the 3' end. So this, you can imagine that this RNA is all compacted together in a way that would allow these template exchanges to occur easily. It's not going to happen if the ends are very far apart. So the reverse transcriptase continues to copy this RNA template, and you can see that's shown here. And as it goes around, it's chewing up the RNA template, except for this little sequence here, the polypurine tract, PPT, which doesn't get degraded by RNase H, and that's because it's going to serve as a primer in a moment. Again, we're making minus strand DNA. We're going all around the genome, as you can see here. And while this is still happening, the enzyme begins to prime a plus strand DNA from this polypurine tract. So this can happen because there are multiple enzymes in the particle, and they can carry out different activities. So now we're starting to make a plus strand shown by the darker blue color, while the minus strand is still being extended. We still have our tRNA on here, it's hybridized, and the initial strand, okay? And now on this panel on the left, the first negative strand has been completed, and we've gone all the way to the end of the genome, to the primer binding site, which is here. And in the process, the plus strand is being made all the way to the tRNA here. The RNase H, of course, is working. It removes this primer right here. And now, and it also removes the tRNA. It has an endonuclease activity that does that. So the positive strand extends to a modified base in the tRNA. Then the tRNA is clipped off. So now you have a single-stranded overhang. And guess what? Right, that base pairs with this end of the negative strand DNA. And the enzyme begins to copy in that direction. So this is the second template exchange, if you will. First one was way in the beginning when that first negative strand went down to the 3' end. So we've made a full-length negative strand. Now we're starting to make the plus strand. Of course, we want to make double-stranded DNA, so that's how that is doing. And finally, we resume at the top here. This is where we were in the last slide. We had a complete negative strand DNA, which was made by copying the RNA with a jump here. And now we're starting to copy back. The primer binding site on the plus strand hybridizes here. Remember, because it's the same sequence on both strands, one is the complement of the other, they can hybridize. So the enzyme then continues to copy this entire negative strand until you have a fully duplex DNA molecule. And this is eventually going to integrate into the host cell. Now let's stretch this out below here just so that we can look at it a little more carefully and see what we've made, because this is where all of this jumping and digestion comes into play. So here is what's going to be proviral DNA once it's integrated. You can see on the left and the right is what we call an LTR, long terminal repeat. This wasn't present in the viral RNA. This has been generated as a consequence of all of these jumps and polymerizations. We have U3RU5. Remember, the U3 sequence was originally present at the 3' end. The U5 was present at the 5' end of the RNA. But look, now it's repeated at both ends. We have U3RU5, U3RU5. And we have the polypurine site here and the primer binding site here. So we've made now a genome, a double-stranded DNA genome with an LTR at both ends. And that's why you do the funny business at the ends. That's why you prime very close to the 5' end and make jumps in order to duplicate these sequences. So in the end, it's really not that complicated. It looks tricky with all of these jumps, but it's really the enzyme starting at an end and by definition running out of template. So it has to jump to the other strand and go around. And then you saw there are a couple of RNA primers that prime this process. We end up with this, a double-stranded DNA, which is different from the RNA. All the genes that encode proteins are the same in here. But the key is we have these LTRs. Now remember, what we had here, for example, RU5 and RU3, now we have U3RU5, U3RU5 at each end.